Hello, in this video I will introduce with some biochemical tests that are performed on microbes to categorize them before genome sequencing. After isolating the microbes from their respective source and evaluating their morphological characteristics like shape, size, color, transparency, and also performing gram staining, it becomes important to do some biochemical tests like starch hydrolysis, MRVP, casein test before the microbes are sent for sequencing to know its novelty. This video is just an introduction to these tests. In the next videos, you will find detailed description for these tests. Biochemical characterization of isolates. Bacteria are classified and identified on the basis of series of biochemical tests. It is a routine test for many group of bacteria, and others are restricted to single family, genus or species. So as you see in this picture, some tests like citrate utilization test, starch hydrolysis, casein hydrolysis. This is just an introduction, so I'll not go in deep. Citrate utilization test is used to differentiate among enteric bacteria on the basis of their ability to utilize citrate as the only carbon source or sole carbon source. Starch hydrolysis test. The ability to degrade starch is used for the determination of amylase production by a microbe. Casein hydrolysis test. This test is used to detect the ability of microbe to degrade the protein casein by producing proteinase enzyme. These tests are very interesting. I will go in detailed description for this test in the next videos. Urease production test. Some microorganisms have the ability to produce the enzyme urease. Indole production test. Tryptophan, an essential amino acid, is oxidized by some bacteria. The enzyme tryptophanase results in the formation of indole, pyruvic acid and ammonia. MRVP tests, that is methyl red Voges-Proveskier test. It is used to differentiate two major types of facultatively anaerobic enteric bacteria that produce large amounts of acids and those that produce the neutral product acetone as end product. Catalase test. This is the test to find that if the microorganisms can produce catalase to break hydrogen peroxide to water and oxygen. These are very simple tests, very easy to perform. I will show in the next videos. So you see these pictures. Starch hydrolysis test. This is the starch hydrolysis test. This is, let me take a color. This is starch hydrolysis test. This is casein hydrolysis test. And these are urease, indole production tests. There are different tests for biochemical characterization of microorganisms.